Have you ever had a sickness that just wouldn't go away until the doctor prescribed you an antibiotic? This was likely because the pathogen in your system, a bacteria, was reproducing more quickly than your body's immune system could fight it. Like humans, bacteria have DNA, genetic information which they use to reproduce. When harmful bacteria begin reproducing within the human body, things get icky. Just as there are millions of types of bacteria, there are several antibiotics that we have invented to combat them. Drugs that can prevent essential life functions of bacterial cells, slow down bacterial growth, or even completely destroy the cells. In this video, we're going to discuss another interesting method of fighting bacteria, preventing them from replicating within the human body.
Before diving into all things DNA, we'd like to first mention the overall purpose of the Huang Research Group. The research discussed in this video is conducted by UW-Madison's Huang Group, led by PI Dr. Xuhui Huang. Their work focuses on using computer models to solve biochemical problems. As stated by the research group themselves, their main goal is to understand and manipulate biomolecular dynamics by developing and applying novel statistical mechanics-based methods that can bridge the gap between experiments and simulations. That's great, but what does that mean specifically for students like us? Behind the complexity of the Markov state model and the generalized master equation model for biomolecular dynamics, big words, I know, it all boils down to gene transcription and protein RNA interactions, which is what me and my LEGO colleagues will discuss today.
DNA is like a script that tells a cell how to properly reproduce, but before it can do that, it must be converted into something the cell can read, RNA. An enzyme called RNA polymerase is responsible for converting double-stranded bacterial DNA into single-stranded RNA. Transcripted strands of RNA serve various vital roles in the overall function of all organisms, including bacteria. Without the process of transcription, essential proteins and functional RNA strands won't be able to be produced, halting bacterial growth.
The protein that aids this process, bacterial RNA polymerase, or RNAP, is highly complex. There are various potential energy functions, equations, catalytic processes, and dynamic conformations that describe RNAP and are essentially needed to holistically describe the Huang's research group. For our purposes, we will simplify the protein as being broken down into three major parts: a rigid base, an area called the beta lobe, and an area called the clamp. DNA fits into the space between the beta lobe and the clamp, and the enzyme can then catalyze RNA creation in a process called transcription. Remember, this enzyme is a protein, which means that it possesses levels of structure. In this case, we are specifically focused on the tertiary structure, or in simpler terms, the 3D structure of protein.
Oh yeah!
A protein is an incredibly dense system whose thousands of molecules are constantly undergoing dynamic conformations. These rotations and vibrations are a natural result of the kinetic energy these particles possess. They are always engaging in submicroscopic random motion.
This fascinating molecule can be used by bacteria to reproduce bacterial cells within the human body. This is what happens when we get sick. So how can we prevent RNAP from completing the transcription process? It turns out the answer lies within the dynamic conformations of the molecule. Kinetic processes drive RNAP to settle into three distinct structures over time: an open conformation, a partially closed one, and a closed one.
This ties into how the mixed antibiotic will bind to a specific site of RNA polymerase, called the switch II site. Because the active site must have a certain shape to interact with the antibiotic, the dynamic nature of RNAP only allows binding when the clamp is partially closed. If the collision between mix and the active site in its partially closed conformation occurs, the molecules will become a complex that effectively inhibits any further movement of the clamp.
Freeze!
When this happens, promoter DNA is no longer able to physically fit between the clamp and the beta lobe of RNA polymerase. This makes the subsequent steps of transcription impossible.
This is the long-term goal of one of the Huang Labs projects, which studies the motion of these moving biological structures and how the mixed antibiotic can interact with them. When mix binds to the switch II protein, various intermolecular forces are formed between the surfaces of the antibiotic molecules and polypeptide structures on RNAP. In this reaction system between conformation selection and induced fit mechanism, delta S is positive because the reaction is forming more complex and dense molecules, which means the increase of microstates and the more flexible ways of energy distributions. In the binding mechanism, the reaction has to overcome entropic penalties, which implies that the protein binding interaction is thermodynamically favorable when the binding occurs. In this reaction system between conformation selection and induced fit mechanism, delta H is negative because the formation of bond between the protein and the mix releases more energy than it is required to break bonds. This makes the reaction overall exothermic. The formation of such electrostatic interaction is an inherently exothermic process. The increasing entropy, combined with the loss of energy and enthalpy, makes the process thermodynamically favorable overall. We can assume these thermodynamic properties because only when the change in Gibbs-free energy is negative can the protein to mix binding occur spontaneously.
While the Huang lab uses computer simulations to model this process, we're going to do it with something much easier: Legos. So here we have our Lego model of RNA polymerase. This pink portion represents the beta lobe and this one is the clamp. And as you can see, these can form all three conformations: the open one, the partially closed one, and the closed one. The hinge here sort of functions as the mobility of the clamp. So we have our polymerase right here, and we have the mixed antibiotic here. And as you can see, we've represented with Legos that it can sort of attach to the switch II binding site, represented by this green Lego, on the molecule. And here we see the mixed antibiotic bind it to RNA polymerase, trapping it in the closed conformation. It's no longer able to open and close freely because the antibiotic is in there, bound and causing a dynamic conformation to the molecule.
In conclusion, using computer models, the Huang group was able to simulate a complex molecular process and ultimately determine a solution to a biological problem. We have seen that the models used in biochemical research are intricate, and the nature of the fascinating molecules that they stimulate can be hard to fully grasp at first. But we have also seen how this information can be easily broken down, just like the individual pieces that make up a Lego set. We hope that you have learned more about RNA polymerase and the mixed antibiotic from this video.